participate. Just put on record. And so she's been just a force here in not only doing excellent science in the marine area, but also being able to mentor and really encourage other underrepresented minorities in STEM education. In my laboratory, she's mentored quite a few Pacific Islanders from Palau, Federated States of Micronesia, and the Marshall Islands. She also has given 2 presentations to the National Academies Committee, one on corals and the present one on sunscreen. And she's been kind of a go-to person on bringing together the best of Western science with traditional Hawaiian knowledge to be able to promote protection of reefs and natural resources. Once again, can I ask people to mute their microphones if you're not Narrissa? And my final comment is that she has been just exemplary in trying to really bring together what we call the so what science. She's passionate about science and the thrill of the chase and understanding new phenomenon. We call her the coral whisperer because she can get corals to do things that some of us haven't been able to do. She's been really important in being able to support UH's mission and making sure that UH is a Hawaiian place of learning. And in her written dissertation, you'll see a number of the oli that she was able to get. And even on spawning night, when our students at Kaneohe Bay waiting for corals.
Dr. Brian Sherry,
people to mute, please. Thank you. She was actually able to give an oli over a cell phone to get the corals to behave, and they did, as a matter of fact, as well. So with no further ado, it's my privilege and pleasure to introduce Narrissa, who will speak to you on her PhD dissertation as part of her defense. Narrissa, take it away.
Good afternoon, everyone. Thank you, Dr. Richmond, for that very long introduction. It's embarrassing, but thank you. Thank you, everyone who's able to join today while I talk about my dissertation work. So I've worked on a lot of things over the past few years, but I'm really excited today to talk to you about these amazing corals that I found that are really resilient to a number of stressors. So I grew up, those of you who don't know, on the Big Island of Hawaii, where I'm presenting from today. And as a kid, I probably spent as much time in the ocean as I did on land. My family's from the Kauai High area, and we're definitely ocean people, like Dr. Richmond mentioned. Much of my work the last decade has focused on corals and how they're affected by human interactions. So today I'm going to talk about my dissertation research, which focused on very resilient coral species and their reproduction and larval settlement, as well as their response to thermal stress. Corals are what truly makes our shorelines unique, and I'm hoping to share a small part of why corals are so important to all of us who call Hawaii home. Corals are my favorite creatures on Earth, and I hope to convince you to love them as well. They might not wag their tails or lick your face, but they're animals. Corals are simple animals, and they share complex relationships with microbes and plants that live inside their tissues. The functional unit of a coral is called a polyp. On corals. Many of them form a colony, and many colonies form a coral reef. Sorry about that. So your average coral reef, when you're swimming, it should be a very loud and bustling place that sounds more like a city than something underwater. There should be snapping shrimp, parrotfish biting and grinding pieces of corals, and so many fish that it looks like rush hour on the H1 going west at 5. Now, swimming over a coral reef, the first thing that most people notice are the fish and the associates, like in this picture. Corals act like infrastructure in the near shore. They're sort of like the apartment buildings of our shorelines, and they act as homes for juvenile fish and allow us to have a vast diversity of ocean life. Coral reefs are the base of the marine food web and support 1/4 of all life in the ocean. Now, most corals start out their lives swimming as larvae, like this one here. They undergo a process called settlement and metamorphosis, eventually dividing and forming a brand new coral colony as pictured at the bottom of this slide. So when you look at all the reasons that corals are important to us, it's staggering that we aren't more familiar with them and that we're not taking care of them as well as we could be. Corals are important for a number of reasons. In Hawaii, coral reefs are big business. People travel here to snorkel and swim, surf on our reefs, which are responsible for clean waves. So if you like surfing, think of coral. Among the most undervalued resources on the planet are the ecosystem services provided by reefs or the intangible benefits provided by their self-built structure. Among the most important is shoreline protection against wave energy and flooding. Corals break up and disperse wave energy that keep our shorelines from eroding. Without coral reefs, damage from large storm events would increase by more than 90%, costing the US $272 billion annually. Coral reefs are also critically important for food sustainability. Many fish we eat are reef dwelling, and some pelagic species spend a portion of their life as juveniles on coral reefs. Recent data also suggests that larval forms of reef fish play an important role in the diet of pelagic fish species, with more than 50% of stomach contents comprised of coral reef fish larvae. More than 500 million people worldwide depend on coral reefs as a direct for their main source of protein, and that alone is valued at more than $1 trillion annually. Corals are also important as a biocultural resource, which I'll talk a bit more about in the next slide. Coral reefs are underwater, so people don't always see the amazing benefits for themselves, and they also don't see when these invaluable resources are damaged or lost. Corals are of great cultural importance, particularly here in Hawaii. The Hawaiian term of ahupua'a, or land division, extends from the mountaintops beyond the shore to the reef crest. We understand that our responsibility for resource protection does not end at the shoreline. Among the best known evidence of that comes from the kumulipo, or the Hawaiian genealogy chant, been passed down from generation to generation and is at least 1,000 years old. It speaks about life emerging from the darkness in our oceans and the foundational unit of life as the coral. To go even further, it recognizes the individual unit of life itself as the coral polyp, or depending on how you translate the word uku ko'ako'a, a coral larva or the process of coral spawning. Unfortunately, this is a poor time in history to be a coral. In the last 40 years, we've lost half the coral reefs worldwide. Corals face a number of stressors, both at the global level as well as the local level. The biggest threat to corals worldwide is climate change. Increased seawater temperatures have already caused massive bleaching events, like the photo in the top left, and the dissolution of carbon dioxide in the ocean is lowering the pH slightly, making it more difficult for corals and other calcifying organisms to maintain growth, while also making them more susceptible to damage from storm events. Local pressures are also affecting coral reef health. Sedimentation, like in the top right photo, is responsible for a large portion of coral mortality in Hawaii each year. Overfishing and herbivore removal impedes the ability for coral species to fend off fast-growing algae, as seen here in the bottom photos. Herbivores, like this parrotfish, act like lawnmowers of the reef. They keep algae levels in check and allow slower-growing corals to grow alongside faster-growing species. One of the major threats to corals that has emerged in recent years is coral bleaching. Corals are animals, but they have a symbiotic algae living within their tissues, just like in this picture here, that can produce as much as 90% of their food, and which also gives them most of their color. When a coral is stressed, such as when seawater temperatures increase, the algae inside the coral tissues start to metabolize a bit differently. They no longer produce food for the coral and instead release oxygen radicals, which can damage coral DNA. So in response to this internal dysfunction, the coral animal expels the algae, exposing the white skeleton beneath its tissue. Now, if the temperature goes back down, coral can recover and recruit new algae back into its tissues from the water column. However, if the temperature remains elevated for more than 8 weeks, coral can actually starve to death. Bleaching also increases susceptibility to diseases and makes it more difficult for corals to fend off fast-growing algae. Bleaching is an incredibly stressful event on corals, and in many cases, the animals will shift their energetic efforts to maintaining health and away from normal biological processes like reproduction and growth. For example, in 2016, following the mass bleaching event on the Great Barrier Reef, it was noted that many of the surviving coral species were just too stressed out and failed to spawn the following year. There have been multiple worldwide bleaching events, which have been increasing in frequency and duration. Sometimes the most resilient individuals come from the least expected places. In 2013, there was a molasses spill in Honolulu Harbor, which was my primary dissertation research site. I'd been studying the reproduction of an uncommon coral species called Leptastria purpurea, found in high abundance within the harbor. When the molasses spill occurred, it released 233,000 gallons into the harbor, killing more than 26,000 fish and countless marine invertebrates, including many corals. Molasses is water soluble, so it couldn't be cleaned up the same way that we'd handle other spills, and instead needed to be flushed out of the harbor naturally, which took over a week. And not only did plumes of molasses block out sunlight, but it also led to a microbial bloom, which depleted the oxygen and caused massive loss of life represented in these photos. The photo of the lace coral colony pictured on the top right here is not bleached as I described in the previous slide. Instead, the tissue just sloughed off and left a dead skeleton behind after being smothered with thick molasses plumes. This large colony was probably over 70 years old at the time of death. Our lab was among the first to survey the area, and what we found was while there was massive loss of life, there were also very resilient individuals that continued to survive even when other animals could not. So Honolulu Harbor, for those who've never dove there, is a very nasty place and is notorious for poor water quality, poor circulation, frequent petroleum, sewage, and chemical spills. It also has a high rate of sedimentation containing excess nutrients, toxicants, and heavy metals, which originate from freshwater sources that empty into the area. There were 2 coral species that dominated the areas we surveyed, and despite the devastating losses of life I saw as a result of the molasses spill, they were thriving in an area where one wouldn't expect to find functional coral reefs. Now, often found side by side, these 2 corals appeared relatively healthy. Leptastria purpurea is pictured on the top right, and a purple coral from the genus Porites is pictured on the bottom right. Based on preliminary analysis, this coral was initially identified as a purple morphology of Porites lobata, which is the dominant reef-building coral in Hawaii. In the presence of these 2 resilient species in Honolulu Harbor was an unexpected find, but we know very little about the coral species in this area, and my dissertation focused on understanding their fundamental biological processes. The first question I posed was, were these animals part of a functional coral reef ecosystem or merely remnant populations that are non-reproductive, basically blinking out? Corals that are under high levels of stress, like those found in Honolulu Harbor, may fail to reproduce, instead shifting energetic efforts towards maintaining health and growth. We saw spawning failures in Australia and Hawaii following bleaching events, and entire reefs of corals in the Virgin Islands are termed zombie reefs, because while they're still alive, no reproduction or recruitment was occurring in the area, essentially making them functionally dead reefs, also known as pseudopopulations. I hypothesized that the harbor corals were what we called zombie reefs that persist in the environment as a result of selection, and that they were non-reproductive. My alternative hypothesis was that they had undergone selection but are reproductive and able to contribute to new recruitment. My second hypothesis was that even if these corals turned out to be reproductive, their larvae would be incapable of recruitment in the highly degraded environment in Honolulu Harbor. My alternative hypothesis was that recruitment can occur despite the high levels of anthropogenic stress present. Lastly, I have hypothesized that corals already under high levels of anthropogenic stress in a harbor environment would not be able to tolerate increased seawater temperatures expected as climate change effects worsen. My alternative hypothesis is, in this case, was that they would show tolerance to stressors that includes increased seawater temperatures. So to address my first hypothesis, I needed to know the basic biology of how coral sexual reproduction works. So there's 2 methods of coral sexual reproduction. The most common form is called broadcast spawning, and that's pictured here on the top. Broadcast spawning corals release large quantities of sperm and eggs. Here's a sperm egg bundle here. And they fertilize externally in the water column. Embryogenesis takes place over hours or days and results in a planula larva that will find a new spot to settle and form a brand new coral colony. About 70% of corals in the ocean are broadcast spawning species, and many spawn only once or twice per year. The other less common form of reproduction utilized by about 30% of corals is called brooding. In this method of reproduction, fertilization takes place internally and a competent planula is released that can settle and form a brand new colony. Brooding corals release fewer planula, but can do so more frequently and can have many cycles per year. Each reproductive strategy has advantages and disadvantages. Broadcast spawning corals, for example, usually have a larger dispersal area and have higher levels of genetic mixing. Larvae resulting from broadcast spawning species have a pre-competency period before they're capable of settlement and spend a longer time in the planktonic phase, increasing their dispersal range. Brooded larvae, on the other hand, have a reduced or absent pre-competency period, and many are capable of settlement as soon as they're released from the parent colony. In certain brooders, this has resulted in limited dispersal, creating genetically isolated populations in some areas. So it turns out that both harbor corals share one other thing in common besides a home address. They are both brooding corals whose larvae contain vertically transmitted algal symbionts. Not only were both species observed to be reproductive within the harbor, but they exhibit seasonal variation in their planulation. Under observation at the Kiwala Marine Laboratory, Leptastria will brood larvae in small quantities throughout the year, averaging 1 to 2 larvae per day. However, in the late summer, during September and October, it has a reproductive peak when a single small colony will release hundreds of larvae per day. Porites lobata is the most dominant coral in Hawaii, and its reproduction has been well characterized as a broadcast spawner. I've nicknamed this coral Harbor Porites because it's not yet been found outside of Honolulu Harbor. Planulation for Harbor Porites appears continuous, and larval release may be tied to physical disturbance in the environment. Due to constraints in permitting with the State Division of Aquatic Resources, we must currently keep this species in a closed system aquarium, which has hindered our ability to monitor reproductive output. My discovery of this brooding coral in Honolulu Harbor was unexpected, and I examined both the skeletal structures and genetics to give us more insight into its presence in the harbor. So Harbor Porites is highly abundant in several areas of Honolulu Harbor. They are encrusting and form small mounding colonies varying from bright purple to green and brown, sometimes present all within the same colony. Within the genus Porites, it's often difficult to positively identify species based on genetics alone, with many species grouping together to form species complexes. To examine the species of sequences of 3 genetic markers, 1 mitochondrial gene and 2 nuclear genes. This data set shows the nuclear ribosomal internal transcribed spacer region, or ITS. The Harbor Porites is shown here in the pink box, grouped closely with the clade 1 Porites lobata species complex, but appears to be distinct. It's also not similar to any of the known brooding species within this genus, which are shown in the green box and also the light blue box. Porites brighami groups into clade 4, and Porites hawaiiensis groups into clade 9. It is possible that Harbor Porites may be a new cryptic species that was hiding in plain sight in Honolulu Harbor, or it could mean that Porites lobata has the ability to shift its reproduction from spawning to brooding based on environmental factors. This highlights the importance of understanding basic functional characteristics of these harbor populations. So since genetically Harbor Porites group so closely with Porites lobata, I examined this coral skeletal morphology. Harbor Porites and Porites lobata look visually similar, although their tissue color differs based on the depth where they're found in Honolulu Harbor. The diagram on the bottom left is an illustration of the skeletal structure inside a Porites lobata colony. The 2 coral skeletons pictured below are genetically verified specimens of Porites lobata. Each round structure highlighted by the red circle is a corallite constructed by the polyp that occupies that area. And the red arrows point to a structure present in the center of the corallite called a columella, which is present in all of these Parietes lobata corallites. So I examined fine skeletal structures in harbor Parietes compared to a type specimen of Parietes lobata. The harbor Parietes skeletons on the top have been dyed using a histology stain so that structures are easier to visualize and photograph. And what I noticed was, depending on where in the Parietes colony the sample was taken, there was variation in the skeletal structure. I took samples from corallites near the edge of the colony and samples from the same colony, but taken from corallites on the top or the center area of the colony. The skeletal sample from figure A was taken near the edge of the colony and is representative of most of the corallites collected from the edges. Of the corallites sampled from the edge of the colony, 54% had a prominent columella as pictured in figure A. On the top left, excuse me, the remaining corallites collected from the edge of the colony displayed a columella that was reduced in height, but still present as seen here in figure B. The coral fragments collected from the tops of the colonies were exclusively made up of corallites that had no visible columella as seen in figure C. The lack of columellas in these samples may indicate reproductive behavior, as this space may have acted as a brooding chamber for coral larvae. Two of my collaborators, Drs. Tisthammer and Forsman, have seen single corallites in genetically verified Parietes lobata samples that lack a columella, but they're usually limited to a single-digit number throughout the entire colony, and they've never witnessed brooding behavior. A more comprehensive genetic analysis is needed to determine if harbor Parietes is in fact a distinct species, as the possibility exists that Parietes lobata is capable of brooding and has utilized this adaptive advantage in Honolulu Harbor. If harbor Parietes is determined to be a new species upon additional genetic examination, we propose the name Parietes halupaensis as its name. This comes from the Hawaiian word halupa, which means to thrive no matter what. So based on the field and lab observations, as well as my skeletal analysis, my null hypothesis of non-reproductive remnant coral populations in the harbor was rejected and instead supported my alternative hypothesis. Not only are both coral species reproductive within the harbor, but high selective pressures within the area have allowed us to characterize what may turn out to be a previously undescribed coral species, or a unique adaptive trait present within Parietes lobata. Leptastria was also not reported as a brooding coral in the literature and has subsequently been identified by colleagues in Guam as brooding there. So I've demonstrated that both corals are reproductive, but we lack the basic understanding of how settlement and recruitment occurs in either species. Reductions in water quality may impede the ability for larvae to detect chemical cues required for settlement. To test if the settlement cues required for successful recruitment are present in the harbor, I designed a series of experiments to determine the settlement cues for both harbor coral species. So for a coral larva to successfully recruit and form a new colony, it undergoes a process called settlement. The familiar line from the Kumulipo accurately describes this recruitment process, beginning with the coral larva. For successful recruitment of coral larvae, there needs to be a suitable substrate with light and water movement. Additionally, a highly specific biological or chemical cue must be present, cues that are often species specific. Once settlement occurs, the coral polyp develops and then calcifies, eventually budding asexually and forming a brand new coral colony. I've performed ecotoxicological experiments demonstrating that anthropogenic compounds, such as pesticides and personal care products like sunscreen, can inhibit coral larval settlement, even at levels determined to be safe by the EPA. With the plethora of anthropogenic stressors within the harbor, it's possible that larvae could be constrained in their ability to settle. So the settlement cue for Parietes larvae was straightforward, very easy. We got high rates of settlement using only a glass biofilm slide that had been suspended in our seawater tanks. We saw high rates of settlement using biofilm slides that were only 10 days old, but the highest rates of settlement were from slides containing pink crustose coralline algae, like is pictured in this photo. The Leptastria settlement Q was more difficult to determine, and various chemical compounds from coral settlement literature were tested at various concentrations without success. A biofilm slide alone induced a low settlement response of approximately 20% in Leptastria. A close observation of Leptastria colonies under the microscope gave clues as to how Leptastria coral larvae recruited to new surfaces. At the Kewalo Marine Lab, where I did pretty much all of my research, I was able to closely monitor Leptastria coral colonies in our open seawater system. And I observed gregarious larval settlement in close proximity to conspecific colonies, or corals of the same species, as well as heterospecific colonies, or colonies of different species. So the photos on the top right demonstrate high levels of settlement along the perimeter of a Leptastria coral colony. Larvae were observed attaching to this area. This is a little larva right here. And within a couple of days, dark brown recruits were observed. And that's what all of these brown spots are in this top photo. So the photo on the bottom right shows how we create biofilm slides in the lab. We basically suspend glass microscope slides in the seawater until they become biofouled, like the one in this well. I determined that larvae reserved in filtered seawater that had previously contained a coral colony induced a settlement response exceeding for 40%. So to recreate that, I created what I called a coral conditioned water settlement queue by incubating a coral colony in filtered seawater for 2 hours, as seen in the photos here on the top left. After removing the coral colony, I added the coral conditioned water to sterile wells along with either a biofilm slide or a plain glass slide to control for spontaneous settlement. I added coral larvae and then incubated the plates overnight in 27-degree ambient temperature and then scored the plates for settlement. So this coral conditioned water queue was first tested in 2014 using different coral species to create a queue. And we hoped that they would settle Leptastria larvae at a high rate. All showed significant rates of settlement compared to the filtered seawater control, and the highest rates of settlement were observed when a queue was created using a Porites lutea colony, and that's what this blue box represents. So I'll be displaying a series of graphs. On the x-axis, we have our queue tested along with a substrate type in some instances. And on the y-axis, we have percent settlement observed after 24 hours. I have repeated this experiment a number of times, in some instances using filters to remove anything larger than 0.2 microns, and it did not have an effect on larval settlement rates. In 2015, we used the exact same Porites lutea coral colony to create coral conditioned water, and again saw a high rate of settlement using Porites conditioned water with a biofilm substrate. In 2016, a year following the massive bleaching event in Hawaii, the exact same coral colony used in previous years no longer induced the high rates of settlement that we'd seen in the previous 2 years, and instead displayed a rate similar to the filtered seawater control here shown in white. Instead, the coral conditioned queue created using a Leptastria colony, shown here in the purple color, displayed high rates of settlement similar to what I'd seen in previous years. And additionally, I noticed that the queue induced settlement at high rates regardless of substrate type, as demonstrated using plain glass slides containing no biofilm. And those are represented on the graph by the pattern here. So after doing that experiment on the 20th, I repeated the experiment on the 24th, 4 days later, using a conspecific colony as well as one of the Harbor Porites colonies I collected from Honolulu Harbor. This time, I saw low rates of settlement across the queues tested. The difference between settlement on the 20th compared to what was happening on the 24th was that I had used a different Leptastria colony to create the coral conditioned water queue. So this series of experiments demonstrated that settlement response to coral conditioned water is dynamic, with the response to our same Porites colony changing over time. And it also demonstrates that there are differences among coral colonies used to create settlement queues, even within the same coral species. In coral populations from Thailand, microbial population shifts can occur quickly as a result of extreme low tides, with colonies displaying altered compositions in as little as 3 days. This may be one method utilized by corals in response to stress, that coral larvae are able to queue in on. If you're a larva and you're in an area with poor water quality, it makes sense to seek out a home where your relatives are surviving. A shift in microbial structure may be a way to determine which coral neighborhoods are better for recruitment at the time of larval release, increasing chances for survival. As Leptastria water showed the highest rate of settlement in 2016, I designed an experiment comparing coral water creating from 6 different Leptastria colonies with varying levels of biological complexity. Leptastria is an encrusting coral and can sometimes be easily removed during collection, while other times a portion of the substrate is collected along with the colony. I created conditioned water cues for each colony pictured and ran a settlement experiment in sterile 6-well plates using either a biofilm or plain glass substrate. And I allowed these to incubate overnight and stored them for settlement over subsequent days. So these are the results of the experiment displayed in these graphs. So we have coral conditioned cues on the x-axis, along with slide types for graph A. Each colony is grouped together by color, and the 2 slide types I tested are displayed side by side. The open bars are the biofilm substrate, and then the pattern bars represent plain glass. On the y-axis, we have percent larval settlement. So what I noticed is that there were differences among treatments. All coral conditioned water cues containing a biofilm were significantly different from the filtered seawater control treatments. But colonies 5 and 6 using only glass slides displayed a rate similar to that of the control. When we examined the settlement regardless of slide type, as in graph B, it became clear that there were differences among colonies used to create the coral water cues. So to try and determine the differences among Leptastria donor colonies used to make the coral water cues, I used ImageJ to assess the colony composition and examine colony biocomplexity. I calculated both the total area of the colony as well as the portion of the colony that was comprised of live coral tissue. The percent of live coral cover varied among coral colonies, but colonies 1 through 4 showed the highest percent of coral cover, all above 80%, as shown in the green text under each colony. So colonies 5 and 6 had the lowest area of live coral tissue, and colony area was high in non-coral biota, including various types of algae. So if you recall the settlement data from previous slides, cues from colonies 1 through 4 induced the highest rates of settlement among coral conditioned water treatments tested, and colonies 5 and 6 displayed an overall lower rate of settlement, similar to that of the filtered seawater control. And when you examine the relationship between the average rate of settlement and the percent coral cover per colony, we see a strong positive correlation between the percent of coral cover, shown on the x-axis, and the average percent settlement, shown on the y-axis. This correlation suggests a direct response to an element of the coral holobiont, which is all the life contained within the coral tissue, and larval settlement. So in other words, as the percent of coral cover increases, so does the average rate of larval settlement in my experiment. Coral conditioned water appears to be unique in its ability to settle Leptastria purpurea larvae. And in fact, the scientific literature suggests that other coral larvae species tend to avoid settlement near adult coral colonies. To verify that this settlement pattern was a phenomenon unique to this species, I used coral conditioned water as a settlement cue for larvae from the broadcast spawning coral Montipora capitata. The water and substrate treatments are presented on the x-axis, and the percent settlement is on the y-axis. We had an overall low rate of settlement for Montipora larvae, but they strongly preferred clean filtered seawater and a biofilm. No settlement was observed among water treatments containing only a glass slide. Similarly, I tested the larvae from a different brooding coral, in this case Harbor Parietes, to demonstrate that coral conditioned water did not induce settlement in all brooded coral larvae. Again, I saw no significant difference among water cues tested, and high rates of settlement only occurred on biofilm surfaces. Only a single instance of spontaneous settlement was observed on a glass slide treatment, validating my earlier experiments that a biofilm is required for larval settlement in this species. These laboratory data suggest a direct relationship between Leptastria larval settlement in the presence of corals, and that it's unique to Leptastria and not a phenomenon observed in other coral species. So as I was reviewing the results of my larval experiments, I saw a very interesting paper that was authored by Danielle Dixon that was published in Science. So she examined the relationship between larval recruitment and chemical cues originating from healthy versus non-healthy areas. So she used a flume system that I tried to recreate in this diagram, similar to one here. So the flume had two sources of seawater, one from an MPA or marine protected area, and one from a non-MPA area that was considered degraded. So the two water sources were used to slowly fill a container at the bottom, where the two would gradually mix. Larvae were placed in the container at the bottom, and their behavior was monitored. So a move towards the side of the container was considered attractive behavior, while swimming away was considered avoidance. So when given a choice, 3 species of coral larvae tested showed that the larvae strongly preferred water that originated from a healthy marine protected area and avoided water that came from a degraded area. Paper also examined the same phenomenon in 6 different reef fish larvae, who also strongly preferred water from a marine protected area over degraded watershed. This graph shows the effect that adding a seaweed chemical cue to the MPA water had on attractiveness. When seaweed chemical cues were present, fish larvae strongly preferred MPA water without the added seaweed cues. A second experiment used water from a degraded non-MPA site, but with added coral chemical cues that would normally be present on a healthy reef. As you can see from this figure, the fish larvae responded positively to the presence of coral cues, strongly preferring non-MPA water with added coral cues. This study demonstrates that larval marine organisms, such as fish and corals, they use chemical cues to determine suitable recruitment habitat. This paper also shows that it's possible for corals to detect these cues, which seem to support my larval settlement results. The next step is to determine if these patterns were applicable in field sites where Leptastria is present in abundance. So I next investigated if these cues were present within Honolulu Harbor, and I wanted to determine if I was seeing a pattern that suggested non-random settlement. Access to areas within the harbor is very limited due to a high volume of boat traffic and inaccessibility from shoreline. So I selected 2 sites where colonies of Leptastria were present as determined in pilot surveys. The first area was directly behind the dinghy dock at Ke'ehi Lagoon, near the boat harbor, close to where you'd launch your boat. And the second site was much larger and located at the western side of the Sand Island beach access area, as pictured on the bottom of this figure. Both survey sites were within sight of each other, separated by a distance of about 1.75 kilometers, and they displayed similar abiotic characteristics. All coral colonies were counted at each site, and measurements were collected for all Leptastria colonies. So the 2 sites differed quite a bit, both in the size and species composition. The Ke'ehi site was a 7 by 2 meter area and contained a high density of corals. I counted 88 Leptastria colonies as well as 734 Harbor Parades colonies. When measuring and photographing Leptastria colonies at the Ke'ehi site, I determined that all but 1 colony was located within a foot or about 30 centimeters of another coral colony. The average nearest neighbor distance measured among coral colonies was approximately 18 millimeters. In the presence of such a densely populated area, I calculated Leptastria population density of 6.3 colonies per square meter. The Sand Island site was much larger in size, but contained few coral colonies. Only 60 Leptastria colonies were counted in the 150 by 100 meter area, and a single Apostolopora damicornis colony was also present. Colonies were spaced much further apart, and less than a quarter of Leptastria colonies were located within a meter of another colony. Those few colonies that were located nearby another coral had an average nearest neighbor distance of 43 millimeters. Despite the larger area available for settlement, the Leptastria population density was much lower at the Sand Island site, with 0.004 colonies per square meter. Leptastria occurs throughout the Indo-Pacific and the Red Sea, but it is always reported in the literature as being less than 5% total coral cover. So to see such a high abundance in the harbor was really unexpected. So with the help of Dr. Amber Wright from the biology department, I used my field measurements to model coral colony distribution at my 2 sites. For the Ke'ehi site, we used size measurements taken from all 88 Leptastria colonies in the field. And additionally, 120 measurements from Harbor Parietes colonies were used to extrapolate sizes from the total 734 colonies that I counted at the Ke'ehi site. For the Sand Island site, all colony diameters were known. And what we did was we ran simulations for both sites 1,000 times each, randomly placing colonies within areas that are identical to the survey size and measuring the average nearest neighbor distance. This figure represents the average nearest neighbor distances calculated for each site from those 1,000 simulations. On the left, we have Ke'ehi, and on the right, we have Sand Island. So at the Ke'ehi site, the average nearest neighbor distance we calculated in our simulation was 39 millimeters, more than twice the observed distance of 18 millimeters that's represented by the blue line. In fact, our observed nearest neighbor distance of 18 millimeters never occurred once in the 1,000 times the simulation was run. And at our Sand Island site, there was only one heterospecific colony and all sizes were known. So the figure on the right side represents our average random means from the 1,000 simulations ran for Sand Island. Since less than a quarter of the colonies at the Sand Island site were within a meter of another coral colony, we didn't have an observed mean as we did with the Ke'ehi site. 95% of our average simulated nearest neighbor distances were greater than 2,079 millimeters. And we predicted that at the Sand Island site, a nearest neighbor distance of approximately 2 meters would be considered non-random. The observed average nearest neighbor distance of 18 millimeters at the Ke'ehi site is just close enough that the Leptastria colony can survive, yet far enough away to avoid competition from faster growing corals. So I was able to reject my original hypothesis that settlement cues for larvae from these species are not present within the harbor. I was able to determine the settlement cues for both Leptastria and Harbor Parietes are present. Harbor Parietes non-discriminately settles on a biofilm surface, while Leptastria larvae show a strong preference for settlement nearby other corals. Our field surveys revealed that in the presence of high coral cover, Leptastria colonies were located an average of only 18 millimeters from the nearest neighbor coral colony. Our model simulations support a non-random distribution of Leptastria colonies, with the observed distance not occurring once in the 1,000 times the simulation was run. This settlement pattern is unique among coral species, as most coral larvae actively avoid settlement near other coral colonies. I could only find one other instance of preferential settlement involving 2 coral species, and that's in the Hawaiian deep sea coral, Kula manamana haumei, which parasitizes bamboo corals by overtaking the host colony. So the last question I had concerning these resilient coral species is how they would respond to increased thermal stress as climate change effects become more apparent and seawater temperatures rise. And this becomes a challenging question to address within the complex system that exists within Honolulu Harbor. Honolulu Harbor is a multi-stressor environment where corals are subject to chronic stress from a number of sources. In 2014 and 2015, during the bleaching event that hit Hawaii, DLNR DAR staff observed 0 bleaching in either species in their facilities at Sand Island. What I wanted to examine was their response to a single stressor, and I chose to examine heat response in a controlled laboratory setting. So for the next set of experiments, rather than using coral larvae, I used coral animals that were fragmented from adult colonies. So these adult colonies were fractured using a hammer and chisel and fixed into underwater epoxy that hardens into a substance that's very similar to coral skeleton. The epoxy is fixed onto the underside of a porcelain tile so that it can easily be moved around. This nubbin creation process is very stressful and damaging for a coral and can take up to 8 weeks to heal properly before nubbins can be used in experiments. After fracture, the Leptastria and Montipora nubbins were placed in seawater tables at the Kewalo Marine Laboratory, where they were allowed to heal in natural sunlight and with particulate fluid that came in with fresh seawater. Because of the constraints of our permit for Harbor Parietes, the coral nubbins created from this species were kept inside the laboratory in a closed system aquarium with artificial cycled light and were fed twice weekly. There was noticeable darkening of the harbor varieties nubbins and colonies, and they went from the bright purple color when the colony was collected in the harbor to a dark brown color. The Leptastria and Montipora coral nubbins were brought indoors 2 weeks prior to experiments so that they could acclimatize to laboratory conditions. A preliminary experiment determined that any temperature exceeding 35 degrees Celsius would kill all 3 species of coral. So my experimental temperatures remained below this threshold. I also matched the irradiance that would be observed under normal field conditions by elevating artificial lights above the treatment containers. I added a third control species, the Montipora capitata, as a species we know is susceptible to bleaching under elevated seawater temperatures. Temperature was ramped up from ambient to the treatment temperatures over the course of 3 hours, similar to what you'd see in a back reef or a tide pool area. And the experiment ran for 20 hours before corals were allowed to return to ambient temperature and then recover in a closed system aquarium at 27 degrees Celsius. I used 4 different temperature treatments ranging from 28.5 degrees to 34.5 degrees. And when comparing photos of nubbins taken at the start of the experiment, I did not note any lightening in color for Leptastria. Coral nubbins were allowed to recover in a closed system tank, and since no mortality was noticed in this species. So since we didn't observe mortality, I determined a bleaching threshold in excess of 34.5 degrees for this species. I also noted that coral polyp mouths dilated with increased temperature during this experiment. You can kind of see that going on here. This is the coral mouth here. The harbor varieties nubbins all showed signs of lightening across temperature treatments. Signs of bleaching were apparent approximately 3 hours post-heat, and these photos were taken 6 hours post-heat. I observed a layer of mucus that formed covering the entire nubbin. You can kind of see like a white sheen on this coral nubbin. That's the mucus coat that was over the colony. So these nubbins were allowed to recover in a closed system tank, and only the 2 lowest temperature treatments survived, placing the survival threshold for this species at 33.5 degrees Celsius. It's important to note that the coral nubbins created from the harbor varieties colonies we collected were kept in a closed system aquarium throughout their fragmentation, healing process, and darkened considerably from the purple color that observed in the harbor. Lastly, the Montipora capitata nubbins were similar in bleaching pattern to the harbor varieties at 6 hours post-heat, with visible lightening occurring at all temperature treatments. Bleaching in this species has been well characterized in literature. Similar to harbor varieties, neither treatment above 33.5 degrees survived. This molecular experiment was similar to the previous one, except there was only 1 temperature treatment at 33.5 degrees Celsius, below the mortality threshold for harbor varieties and Montipora capitata. I placed replicate tanks within a single large water bath. As with the previous experiment, water temperature was ramped up from 27 degrees ambient to 33.5 degrees over the course of 3 hours, and monitored with data loggers measuring temperature and light every 60 seconds. At the start of the experiment, 3 hours post-heat and 20 hours post-heat, a coral nubbin from each species was flash frozen in liquid nitrogen to preserve molecular expression. Coral samples were pulverized and protein was extracted and quantified. 60 micrograms of protein was loaded into acrylamide gels and separated by size. Proteins were transferred to a membrane and blotted with antibodies to examine protein expression. So I examined the expression of a protein called superoxide dismutase, specifically SOD1. SOD is a molecule that is important in limiting the effects of damaging reactive oxygen species that can damage DNA and that are created in coral tissues. Under thermal stress, levels of oxygen radicals increase and can damage cells and signal the start of apoptosis or controlled cell death. The graphs on the left are Western blot intensity values for SOD at each time point collected. My treatments are on the x-axis and the signal intensity is on the y-axis. The Leptastria samples in our experiment showed no signs of bleaching at 33.5 degrees. The control samples had low levels of SOD expression that increased with temperature over time. Our harbor variety samples showed a high level of expression in the temperature controlled samples, a decrease at 3 hours post-heat, and an increase in expression at 20 hours post-heat. The Montipora capitata samples displayed a similar expression pattern to that of the harbor parietes. Both harbor parietes and Montipora showed signs of bleaching at 3 hours, and these photos were taken 6 hours post-bleaching, post-heat, to show how similar the bleaching patterns were overall compared to Leptastria, which did not bleach. One observation I suspect may be contributing to the differential expression and bleaching patterns is the presence of a pink fluorescent pigment in the Leptastria samples. The presence of this pigment was the only noted in Leptastria, and it was really frustrating when it happened because no matter how we tried to get rid of it, we couldn't get it out of the sample. And it was present in Leptastria, but not in the other two species. So many corals contain fluorescent pigments, and the protective benefits of these pigments have been well documented in scientific literature. It's unknown if the coral animals themselves produce these pigments or if they're produced by algal symbionts and sequestered in the coral tissues. They display antioxidant properties and are shown to reduce light penetration in coral tissues, leading to an overall reduced temperature rate inside of the coral as detected using microsensors in other coral species. The photos on the top right show Leptastria polyps under ambient light as well as under UV light. You can clearly see the pink fluorescent coloration throughout the polyp tissue, likely the source of our pigment seen in the extracted protein samples. There's also a concentration of green fluorescent pigment around the polyp mouths that is very obvious. In our temperature treatments, I observed the dilation of Leptastria polyp mouths in response to increased temperature, as seen in the photos on the bottom of the slide. Harbor parietes displays a strong fluorescent signal in samples freshly collected from the harbor. Again, one of the constraints of our permit was that we were required to keep the colonies in a closed system indefinitely. And after several months with artificial light, the fluorescent properties in harbor parietes were no longer visible. I feel this contributed to the lower bleaching and survival threshold for harbor parietes in my experiments, which seems to contradict the reports of zero bleaching in this species observed during the thermal event that hit Hawaii in 2014 and 2015. So the results of my thermal tolerance experiments revealed that while Leptastria performed phenomenally compared to harbor parietes, both species display thermal tolerance above the average temperature range recorded nearby. I rejected my null hypothesis and determined that under conditions in the field, both corals are thermally tolerant. The multi-stressor environment in the harbor does not reduce the ability for coral populations to withstand thermal stress. We're living in a world that's changing very rapidly. The effects of climate change are becoming more apparent as we lose coral reefs that are so valuable to life in the ocean. There are lessons to be learned about these two harbor coral species. They're not zombie corals, but functional and reproductive animals that display high levels of resilience despite poor water quality and circulation within the harbor. Through collaborative efforts from my colleagues, we've discovered a lot about their reproductive strategy, survival mechanisms, and distribution. My dissertation research provides foundational insights into these unique coral populations and puts these two species on the radar for researchers and managers who will hopefully continue to study these important coral species. I mentioned earlier the cultural understanding that coral larva was the most basic unit of life that forms in a coral colony. This single line from the Kumulipo, a genealogical history more than 1,000 years old. This one line of text was the inspiration for my entire dissertation and 4 individual research chapters. Imagine how much other knowledge was known to our kupuna and awaits recognition under a Western science lens. If you examine the original translation of this Kumulipo line by our late Queen Liliuokalani, you see that the word uku ko'a, whose modern translation is interpreted as coral larvae, can also be translated into coral insect. Perhaps my kupuna were onto something in their observation of our coral reef ecosystems. So people often joke that the last 2 things we'll find on Earth after a nuclear holocaust are Twinkies and cockroaches. These coral insects, or cockroach corals as I lovingly call them, possess all of the positive properties of the unofficial bird of Hawaii. They're resilient, they persist, and we find them in places deemed inhospitable towards most living things. Through my dissertation experiment, I've identified corals that we know are going to be good candidates for further scientific studies, as well as cultivation and hopefully transplantation. We need to get these cockroach coral populations that survive in marginal environments onto researchers' radars to further understand how they're able to survive so that we can apply those tools to other less resilient coral species. And before I take your questions, I'd like to take a couple of moments to thank people who've been invaluable in helping me complete my research. Each of my collaborators and field support colleagues gave me their time and knowledge to make my research possible, including a few brave souls who dove in the nasty waters of Honolulu Harbor with me. Thank you, James and John, for boldly going into the harbor waters with me. I'm also very appreciative of the mana wahine, or mentors, that have guided me throughout the years, encouraging me to expand my knowledge and attend graduate school. I fought it at first, but they sure taught me. None of the work in my dissertation would have been possible without support and funding from a number of entities. Most of these I've added here, but I'm sure I'm missing some. This page looks a lot like NASCAR sponsor logos with all the support that I received. And I would particularly like to thank the Kohala Center and their funders for awarding me the Mellon Hawaii Doctoral Fellowship, which allowed me the time and the funding to write my dissertation. I'd also like to acknowledge and thank my dissertation committee members who gave me their time, energy, editing skills, and brilliant minds to help mold me into a better scientist. There have been some changes to the roster over the years, but each of you has played an important role in my growth. Thank you all for your support and encouragement. Throughout this PhD journey, I've met some truly amazing friends and colleagues. Each new lab, office, or group that I joined, I connected with people who made this entire process both tolerable and enjoyable. And if it wasn't for you guys, your encouragement and listening ears, I don't think I would have made it to the end of this journey. I'm so grateful to each and every one of you. The other steady constant in my life has been my family. I'm blessed to be able to have people around me who love me unconditionally, and no matter what mood my dissertation might have put me in. Thank you for your love and patience over the years, and hopefully soon I'll be in a better mood and better equipped to answer questions like, when are you graduating and are you done yet? Lastly, I'd like to give a special thanks to my PhD advisor, Dr. Bob Richmond. At the start of my degree, I thought I'd just be doing a PhD, but it was also sort of a postdoc or two rolled into one. I learned so much about being a scientific communicator, as well as the relationship between science and policy. Now, pictured in this slide are the most 6 terrifying moments in my life, all of which occurred in the last 5 years, each more terrifying than the last. The first being asked to present in front of the White House Council on Environment and Quality when I was an advocate for the expansion of Papahanaumokuakea. The second, when I was asked to speak at the United Nations on the importance of marine protected areas and conservation. The third was presenting my research and talking about the Kumulipo in front of Auntie Pua Kanahele, who was awesome, by the way. The fourth was when I did a story collider about using my voice to communicate about science in front of a live audience that would eventually be broadcast nationally. The fifth was when I was the poster child and presenter at the SACNAS Diversity in STEM conference, where I gave multiple talks and addressed a crowd of over 5,000 people in the most unique and epic Halloween costume I've ever had. And lastly, I got the opportunity to speak on NPR's Science Friday about resilient corals and their importance in a changing climate. Dr. Richmond, none of this would have been possible without your encouragement and my public speaking development while in your lab. I don't think I'll ever be able to capture with words or photos how important that you've been in my development as a scientist and human being. Thank you. That's the end of my thank you slides. Please enjoy this picture of a Leptastria colony that made poor life choices and is now surrounded by a Harbor Parades colony. Thank you for listening to me speak, and I'm happy to answer any of your questions.
Okay, feel free to unmute.